Hello dear students, today is our lecture Antibodies, Immune Response, Immunopathological Reactions, Immunoprophylaxis and Immunotherapy. The origins of the doctrine of antibodies and their protective role can be traced back to the outstanding scientists. In 1885, Paul Ehrlich first proposed the theory of humoral immunity, for which he received Nobel Prize. In 1887, Behring and Kitazada developed the first antitoxin serums for diphtheria and tetanus exotoxins. In 1959, Porter and Edelman deciphered the chemical nature of antibodies, and they shared the Nobel Prize in 1972. In the second half of the 20th century, thanks to the research of Burnet and Tonegawa, the nature of the specific diversity of antibodies and the genetic basis of this phenomenon were studied. They also received Nobel Prize in 1960 and 1987 accordingly. So let me remind you that we have two main types of the immunity. Individual immune response consists of non-specific or in nature first line of defense and specific adaptive or acquired immune reactions. The adaptive immune system, I also called acquired immunity, uses specific antigen strategically. The adaptive immune system, also called acquired immunity, uses specific antigens to strategically mount an immune response. Unlike the innate immune response, which attacks only based on the identification of general threats, the adaptive immunity is activated by exposure to pathogens and uses an immunological memory to learn about the threat and enhance the immune response accordingly. The adaptive immune response is much slower to respond to threats and infections than the innate immune response, which is primed and ready to fight all the times. And if you remember, there are PRI receptors, two pumps. There are two main mechanisms of immunity within the adaptive immune system: humoral components and cellular components. Humoral immunity is also called antibody-mediated immunity. With assistance from helper T cells, B cells will differentiate into plasma B cells that can be produce antibodies against a specific antigen. In case with cellular immunity occurs inside infected cells, and this immunity is mediated by T lymphocytes and also called cell-mediated immunity. So, we need to know that, as you see, the adaptive immune system is made up of T lymphocytes. In our case, we have different types of T lymphocytes, B lymphocytes, as you see, and also antibodies, antibodies in blood and other body fluid. T lymphocytes produce in bone marrow, and then they move to thymus through the bloodstream, where they mature. T cells have three major functions based on their phenotypes. You see, we have only two phenotypes in this case. In case with T helper cells, CD4 plus cells, they use chemical messengers to activate other immune system cells in order to start the adaptive immune system, because they called T helper cells. They help to activate immune system. They are like vector for activation. In case with cytotoxic T cells, here CD8 plus cells, they detect cells infected by viruses or tumorous cells, for example, and destroy them. They don't need some helpers for activate immune response. They can directly lysis these infected cells. Sometimes some T helper cells become memory T cells after the infection has been defeated. They can remember which germs were defeated and are then ready to activate an adapted immune system quickly if there is another infection. This is called immune memory. T cells have detection features on their surface that can attach to germs like a lock that one particular key will fit. We described this more because it's specific immunity and they require some features. The immune system can produce a matching T cells type for each germ is an infection within a few days. Another type of cells for adaptive immunity, another type of lymphocytes, they called B lymphocytes, B cells. They are made in the bone marrow and they mature there to become specialized immune system cells. Like T cells, there are many different types of B cells that match particular germs. The B cells are activated by T helper cells. They need some helpers for activation. Helper T cells then release cytokines, and that leads to the proliferation and differentiation of the B cells into plasma cells, and plasma cells secrete antibodies into the lymph and blood to target the specific pathogen. And the last components of adaptive immunity, this is humoral components of the adaptive immunity, very specific components, and this is components we describe more in today's lecture. This is antibodies. Antibodies are compounds of protein and sugar that circulate in our bloodstream and specifically target invading pathogen and excel the neutralizing bacteria and viruses present in the body fluids or extracellular space. Here are the complex mechanism of our adaptive or specific immunity. Like we talked before, we have cell-mediated immunity and antibody-mediated immunity, humoral components and cellular components. To participate in an adaptive immune response, naive T cells must first encounter antigen. We have in this case T0 lymphocytes profile, and then be induced to proliferate and differentiate into cells capable of contributing to the removal of the antigen. These T0 phenotype cells also need some activated stimuli. Immature T cells are activated to effector T cells after coming into contact with antigen-presenting cells. Let me remind you, we have some different types of antigen-presenting cells, and the most important, this could be macrophages and also dendritic cells. Antigen-presenting cells, during these encounters, antigen subparts or peptides with a protein base are placed into contact with the major histocompatibility complex of the cell. This means this is cell complex and receptor complex. Other antigen-presenting cells on their surfaces have MHC molecules, but they have different types of MHC molecules. This can be MHC1 or MHC2 molecules. And these complexes then presented to peptide receptors or on T cells, TCR receptors. These T cells may be from one of several classes. In case with MHC2 receptors, we activate T helper cells. In case if here is active MHC1 receptors and peptide and MHC1 receptors, we activate T0 phenotype cells to cytotoxic T cells, CD8 plus cells. The classical mechanism when the pathogen's antigen are expressed on the cell surfaces or in antigen-presenting cells. Helper T cells release, some cytokines that can activate the cells bind to the infected cells MHC antigen complex and differentiate into cells into cytotoxic T cells also. The infected cell then undergoes lysis. This is main mechanism of cytotoxic T cells. Antibody mediated immunity represented more on this slide. With assistance from T helper cells, like you see here, we first activate T helper cells, and then T helper cells can activate B cells. With assistance from T helper cells, B cells will differentiate into plasma B cells that can be produced antibodies against a specific antigen. The humoral immune system deals with antigens from pathogens that are freely circulating or can be outside of the infected cells. Antibodies produced by B cells will bind to antigen, neutralizing them, causing lysis or phagocytosis. This is the best example in immunology of collaboration between humoral and cell-mediated immunity, like you see in this slide. Firstly, we activate cell-mediated type of immunity, and next, cell-mediated type of immunity collaborate with antibody-mediated immunity to producing specific antibodies against specific antigen. Let's focus more of antibodies. Antibodies or immunoglobulins, large gamma globulins that specifically target invading pathogen. Antibodies are produced, like we said, by plasma cells in response to infection, or it can be also immunization, and bind to and neutralize pathogens or prepare them to uptake for phagocytosis. Each antibody molecules has unique structure that enables to bind specifically corresponding antigen, but all antibodies have the same overall structure. The components here you see of the immunoglobulin G molecule characterized by heavy and light chains connected by disulfide bonds. Each chain consists of variable region, variable domain, and constant region, constant domain. The variable regions are lightly and heavy chains form a binding site for antigens. Here we have antigen binding site. The two N-terminal fragments are called the Fab region. Here, if we describe more, you can separate antigen structure by two components. It could be easy for you. Two Fab fragments, and the antigen, like in Fab, stands for antigen binding site. Like we said before, about variable and constant variable regions of heavy and light chains form is this antigen binding site. In other case, we call this Fab fragment. The C in Fc stands for crystallize. This is Fc fragments because the well-conserved amino acid sequence allows this fragment to crystallize. Also, by the way, Nobel laureates Potter and Edelman of leading importance in the study of the structure of antibody molecules. They used papain, some pepsin, and plant protease in the presence of cysteine, which destroys disulfide bonds. Papain splits the immunoglobulin molecule into three fragments. You see this one. This was here experiments. Two Fab fragments and one Fc fragments, like we said before about disulfide bonds. And here they break these disulfide bonds. Also, you can use another type, pepsin, and pepsin cleaves immunoglobulin G heavy chains at the point below the disulfide bond. Yeah, and because of this, that resulting fragment consists of two Fc fragments linking together. You can separate this by pepsin, and also one Fc fragment. Okay. Here are 5 classes of immunoglobulins. The classes of antibody molecules are also called isotypes, and they are named by heavy chains. Immunoglobulin G, gamma heavy chains, immunoglobulin M, mu heavy chains, immunoglobulin A, alpha, immunoglobulin D, delta, and immunoglobulin E, epsilon. Immunoglobulin A class consists of monomeric or dimeric forms. Immunoglobulin M here pentameric form, and also immunoglobulin G, immunoglobulin E, immunoglobulin D are monomers. The properties of immunoglobulins of different classes are not the same. Like you can see on this slide, the participation of antibodies of different classes in immune reaction is also different. Main function of antibodies, like we said before, this is recognition, connection, neutralizing of antigen, the most important function. Also, antibodies can help in antigen presentation to macrophages and antigen-presenting cells. Also, they can activate of complement and lysis of cells containing foreign antigen. Also, they can have a lead role in antibody-dependent cellular cytotoxicity, and also they can have a role in opsonization of corpuscular antigen and attraction to phagocytosis. Attraction some macrophages to phagocytosis. For example, for instance, here during the phagocytosis, during this opsonization function, antibodies bind to bacterial surface antigens, and these opsonins can lead to activate phagocytosis cells. They can light these phagocytosis cells. And additions, macrophages may process and present protein antigen to specific T cells during a primary immune response. This is connection between innate immunity and adaptive immunity. Let's start talk more about specific function of each isotypes of immunoglobulins, and we start with immunoglobulin M molecule. Among all immunoglobulins, because the immunoglobulin have pentamer structure, immunoglobulin M molecule has highest molecular weight. It is present only in intravascular compartments, but not in body fluids or secretions. Immunoglobulin M usually exists in these pentameric forms. 5 immunoglobulin monomeric units, like this one, tails, joined with each other by G chain. Here G chain to form a complete immunoglobulin M pentamer molecule. This immunoglobulin has 10 Fab fragments. You can see one immunoglobulin M have 10 Fab fragments. Because of this, immunoglobulin M is the first antibody to be produced following infection, like primary immune response, and they represent acute or recent infection. It is also called primary immune response antibody. Immunoglobulin M is also present on B cell surface, like monomeric form, and serves as a B cell receptor for antigen binding. And this is understandable why immunoglobulin M molecule have the leading role in primary immune response, because if you return our presentation to B cells activation, you see here our B cell has some receptors, and these receptors can be immunoglobulin M receptors, and these B cells is activated by T helper cells, and these B cells can produce, they develop in plasma cells, and plasma cells produce antibodies, and because of this they have receptors of immunoglobulin M molecule. Because of this we see that first molecules, immunoglobulins, they can produce in primary immune source immunoglobulin M. So next one, immunoglobulin G molecule. Among all immunoglobulin, immunoglobulin G has maximum daily production, longest half-life of, for example, 28 days, and highest serum concentration. This means that they can be present a lot in our secretion and our blood stream. Immunoglobulin G has 4 subclasses, and this is important: immunoglobulin G1, immunoglobulin G2, immunoglobulin G3, and immunoglobulin G4. Immunoglobulin G also can cross placenta. This is unique mechanism for immunoglobulins. Hence, immunoglobulin G can provide immunity to the fetus and newborn. Among subclasses, immunoglobulin G2 has the poorest ability to cross placenta. Many of the effector functions of antibodies are mediated by Fc fragments of immunoglobulin molecules, and here. And for instance, some immunoglobulin G subclasses, in this case immunoglobulin G1 and immunoglobulin G2, bind to phagocytosis Fc receptors and promote the phagocytosis of antibody-coated particles. Some subclasses of immunoglobulin G, like immunoglobulin G, immunoglobulin G2, has limited extent, activate the complement system. Immunoglobulin A, the second most abundant class of immunoglobulin next to immunoglobulin G, constituting about 15% of total serum immunoglobulin. Like we said before, there are two forms presented of immunoglobulin M. Immunoglobulin A in serum predominantly exists in monomeric form. Serum immunoglobulin A interacts with the Fc receptors expressed on immune effector cells to initiate various functions such as antibody-dependent cellular-mediated cytotoxicity. It also can be degranulation of immune cell secretory immunoglobulin G, immunoglobulin A, is dimeric in nature, and this consists from two monomeric form of immunoglobulin A. And also these two immunoglobulin A monomeric units can't be joined by D chain. In addition, this is another joint segments present between two immunoglobulin M monomeric molecules and also called secretory component. The dimeric secretory immunoglobulin A is synthesized by plasma cells situated near mucosal epithelium, but D chain is also produced in the same cells, but secretory immunoglobulin A mediates local and mucosal immunity because of this, provides protection against pathogens by cross-linking bigger antigen with multiply epitopes and preventing the entry through the mucosal surface. And because of this, we see that major secretory in mucosal immunity of this. Immunoglobulin D is found as membrane immunoglobulin on the surface of the B cells and acts as B cells receptor along with immunoglobulin M molecule. But in this case, not as the function is known for immunoglobulin D so far, so because of this we can describe this immunoglobulin more. And the last one, immunoglobulin E. Among all immunoglobulins, immunoglobulin E is having the lowest serum concentration, shortest half-life, and minimum daily production. It is also the only heat-labile antibody. This antibody you can inactivate at 57 degrees in 1 hour. Immunoglobulin E, despite this all, immunoglobulin E is highly potent and mediate type 1 hypersensitivity reactions by binding to the mast cell, causing their degranulation. Immunoglobulin E response is seen in various allergic conditions such as asthma, anaphylaxis. Also, immunoglobulin E elevated in hemetic infection. By coating on the surface of eosinophils, immunoglobulin E stimulates release of the mediators onto the surface of helminths by the process of an antibody-mediated cellular cytotoxicity. Very interesting roles of this immunoglobulin A against parasitic infections. So, like you see, immunoglobulins have a protein nature. Therefore, when introduced into the body, they show antigenic properties. Based on the location of antigenic determinants, the immunoglobulin molecules are divided into isotypes. Here we described these 5 isotypes. Also, they can be divided into idiotypes, allotypes. Allotype antigenic determinants are individual. They distinguish the immunoglobulins of one person from another in terms of antigenicity. Idiotypic antigenic determinants reflect to the peculiarities of the structures of the variable part of immunoglobulins. That is, they depend on the antigenic specificity of idiotypes and idiotopes. Many structural features of antibodies are crucial for their roles to their ability to recognize antigens and to their effector functions. And the interaction of epitopes of antigenic with paratopes, active centers of antibodies, is characterized by affinity and avidity. You see, yeah? Increased affinity, the functional significance and the functional properties. Affinity, this is the strength of the specific interaction between antigen and antibody. And this affinity depends on the degree of spatial correspondence between the epitope and paratope. And also, in additional of affinity, avidity, another important concept in the interaction between antibody and antigen there is specificity. Also, B cells begin to produce antibodies other than immunoglobulin M antibodies. This process is called heavy chain isotype class switching, or immunoglobulin class switching, or class switch recombination. This is a biological mechanism that change B cell production of immunoglobulin, for example, isotype M to isotype G. The genetic basis of class switching and antibody diversity is a complex and really fascinating area of the research. Thanks to Suzumu Tonegawa, we know this process for today. This is gene-determinate processes and pretty tricky mechanism. So if you want to learn more and to know more about this, I recommend to you read Abba's Cellular and Molecular Immunity book, and also here example of this antibody class switching by VDG recombination. And I need to mention that Suzuma Tonegawa received the Nobel Prize in 1987 for his discovery of the genetic principle for generation antibody diversity. Next, our slide about different types of antibody response. We mentioned before about immunoglobulin M start to produce firstly, and this is called primary antibody response. So we need to learn more about this because each immune reaction develops in stages. First, because this is adaptive specific immunity, they need to formate immune complex, or in other case, this is called like immune synapse. And first stage, this is recognition of the foreign antigen by lymphocytes. Next, this is activation of lymphocytes with their proliferation, formation of specific clone, synthesis of cytokines, and specific antibody. Like we mentioned before about cell-mediated immunity and antibody-mediated immunity, about first step of activation and the last exit of this activation. Primary antibody here and secondary antibody response demonstrated here. Primary responses result from the activation of previously unstimulated naive B cells, whereas secondary response are due to the stimulation of expanded clones of memory B cells or T cells. Phases of the primary immune response are the next here. Log phase, log phase, exponential phase, plateau phase, and decline phase. Late phase, or also called latent phase, and this is about 3 or 5 days. Exponential phase 7-15 days. This is the phase of the rapid increase of antibody production by plasma B cells. The increase in antibody production is because of the large number of these plasma cells. And the plateau cells can be almost 1 month to 30 days, and phase of reduction or decline phase of antibody synthesis can lead to half of the year. The secondary immune response, you see, not have all these phases, and they compared to them, they have absolutely different antibody amounts. And the secondary immune response has a very short this lag phase, latent phase, you can almost see them. Which means that both lag and exponential phase take place at go. The antibody production levels increase rapidly without a short period, normally within a few days. Compare, please, 7 days and 3 days. This is because of the antigen-specific memory T and B cells produced during the primary response. Features of this secondary immune response the next. Short latent period, intense, like powerful reaction of antibody formation, antibody amount, immunoglobulin G synthesites can be first, and therefore the secondary response develops more rapidly than the primary response, and largest amount of antibodies are produced in the secondary response. So, we have previously mentioned that effector functions of antibodies require the binding of heavy chain C regions, which make up for Fc portions to other cells and plasma proteins. For example, another features of these immunoglobulin Ls, M, G, E, A, that immunoglobulin G coats microbes and targets them for phagocytosis by neutrophils and macrophages, like opsonins. Another their mechanism. This occurs because the immunoglobulin G molecule is able to stimulatively bind through the FAP region to the microbe and through the Fc region to immunoglobulin G heavy chain specific receptors that are expressed on neutrophils and macrophages. In contrast, immunoglobulin E binds to mast cells and triggers their degranulation. This is not some, specific mechanism of antibodies, and in case with immunoglobulin E, they activate mast cell degranulation because immunoglobulin E and mast cells can express immunoglobulin E specific Fc receptors. Another Fc-dependent effector mechanism of humoral immunity is the activation of the classical pathway of the complement system. Like we also said before, different subclasses of immunoglobulin G molecule. The system generates inflammatory mediators and promotes microbial phagocytosis and lysis. It is initiated by the binding of the complement proteins called C1q to the Fc portions of antigen complex immunoglobulin G or immunoglobulin M molecules. The Fc receptors and complement binding sites of antibodies are found within the heavy chain C domains of different isotopes. But you can say, based on this slide, the main function of this immunoglobulin, this is against different types of infection. And also we have some inappropriate in case features of this immunoglobulin, like immunoglobulin E molecules can simultaneously activate mast cells degranulation, and this can lead to immediate hypersensitivity type 1. But in case with immunoglobulin M, this is major immunoglobulin in primary immune response. Immunoglobulin G subclasses can lead to opsonization, phagocytosis, complement activation, neonatal immunity, and so more. In case with immunoglobulin A antibody, this is about big mucosal immunity, because through epithelia we have the most large amount of secretory immunoglobulin A. So you see, this is really a unique mechanism for all immunoglobulins, but they is based in specific immunity. In case of isotype switching, this is called antibody-mediated specific immunity. We repeatedly emphasize that unlike innate immune responses, adaptive responses are highly specific to the particular antigen that induces them. Because of this, you see antibodies have unique features. An individual is capable of making tremendous number of structural distinct antibodies, perhaps in order of millions, each with a distinct specificity. The ability of antibodies in any individual to be specifically bind a large number of different antigens is a reflection of antibody diversity. Antibodies can be remarkably specific for antigens, distinguish between small differences in chemical structure, like antigen A And we have two types of antibody, antibody B and antibody A. And only in antigen B, only antibody B can connect with this antigen. For example, FAB regions of antibody B, of antibody A, can't connect with antigen B. This fine specificity of antibodies applied to the recognition of all classes of molecules. The concept of non-cell discrimination with this ability of the immune system to recognize and respond to foreign antigens but to not self-antigens. Sam McLaren-Burnett, among the first to hypothesize clonal selection, added the corollary that lymphocytes specific for self-antigens are eliminated to prevent immune reactions against one's own tissues. The phenomenon was called immunological tolerance. Based on the discovery and study of this phenomena, Medawar and Burnett received the Nobel Prize in 1816 for opening acquired immunological tolerance. Elucidating the mechanism of self-tolerance is the key to understanding the pathogenesis of autoimmunity. Definition of tolerance can be like absence of immune response in the presence of antigens acceptable to lymphocytes in the body. In other words, it's the ability of the immune system to distinguish between self-antigens and foreign antigens. Features of immunological tolerance, it is caused by substances of antigenic structure, tolerogens. It appears specifically to certain antigens that induced its formation. The duration and degree of manifestations vary greatly. Our body responds to various pathogens such as bacteria, viruses, production of a wide variety of antibodies that can bind to specific antigens. This is called the diversity of antibody. Burnett's idea that specificity for diverse antigens exists before these antigens can encounter our body. This means that we create a lot of antibodies before this antigen can enter our body. The clonal selection theory proposed that antigen selects lymphocytes for activation from a population of cells pre-committed to produce specific antibody. This means antigen choose the naive lymphocytes, not T lymphocytes choose antigens. Nowadays, this theory is not so classical. But implicit in this theory is that antibody-forming cells are monospheric, like different receptors for specific antigens, and express cell surface receptors capable of binding foreign antigen, unknown foreign antigen. These are several mechanisms of tolerance, which can be broadly categorized as either central tolerance or peripheral tolerance, and this is more unique modern definition of immune tolerance. Central tolerance prevents the maturation and egress of autoreactive immune cells, for example via clonal deletion of T cells in thymus. Any autoreactive autoreactive cells that escape central tolerance, for example, can be induction of energy or suppression by mechanism of action of regulatory T cells. I want to add that induction of immunological tolerance is possible therapeutic strategy for preventing harmful immune response. This is great interest in inducing tolerance to treat autoimmune, allergic disease, and to prevent the rejection of organ transplant in clinical trials now under the way. Also, we previously mentioned about specificity. However, some antibodies produced against one antigen may bind to a different but structurally very similar related antigen. This is referred to as a cross-reaction of antibody specificity. Antibodies that produced in response to microbial antigen sometimes cross-react with self-antigens, other antigens, and this may be the basis of certain immunological disease. Autoimmunity results from inadequate self-tolerance or regulation of T lymphocytes. Autoimmune, this reaction may be triggered by different stimuli, and nowadays we not know all these stimuli. And in case when you break this immune tolerance, this breaking of tolerance leads to autoimmune reactions, and in case of the immune reaction, we are developed some autoimmune diseases. The exact, like I said, the exact cause of the immune disorders are unknown. Many autoimmune disorders have been strongly associated with genetic, infectious, or environmental predisposing factors. The fundamental underlying mechanism maybe of autoimmunity is defective elimination and control of self-reactive lymphocytes. Autoimmune disease vary greatly in the organs that affect, and the clinical manifestation, with some being limited to particle tissues and other being systematic or dissemination. Also, in case with inflammation process, this can lead to exposure of self-antigens that are normally concealed from immune system. Despite this variation, all autoimmune diseases are believed to go through sequential phases of initiation, propagation, and resolution. In this slide, you see the most common autoimmune disease. In the conventional autoimmune diseases, the tissue distribution of the autoantigen to a large extent determines whether the disease can be organ-specific, like this one, or non-organ specific. For example, this is lupus erythematosus here, and in case with lupus erythematosus, which is excellent example of systemic autoimmune disease, some protein against some protein can lead to develop some antinuclear antibodies, and these antinuclear antibodies can be present interact with the nucleus of all other cells, and the lesions are not confined to any one organ. And pathological changes are very different, widespread, and they're seen in the skin, and because this disease called lupus. Lupus butterfly rash on the face can be specific symptoms of this disease, but it also can lead to systemic inflammation and systemic damages like kidney glomeruli, joints, serous membranes, blood cells, blood vessels. A variety of these human diseases are caused by immune responses to non-microbial environmental antigens. This means maybe based on tissue damages, we exposure a lot of self-antigens, and based on their like this one, anatomic alteration of this tissue, we have systematic autoimmune disease, or in case with kidney, post-streptococcal glomerulonephritis can be like organ-specific autoimmune disease. Also, let me remind you that infection I dealt with appropriate immune responses that detect foreign antigens. Another example of this, when in case with adaptive immune responses, the necessity for clonal proliferation of lymphocytes in order to generate sufficient numbers of antigen-specific cells. And here regulate processes, and because of this, antibody of class appropriate to clear the infection is produced and bind to the surface of the pathogen. In the regulation of the immune responses, failures are possible. And these failures can lead to another type of immune disorders. This immune disorders called hypersensitivity disorders. In case with autoimmune disease, this is exposure of cell antigens, breaks of the immune tolerance, and in case with immune response, specific immune response, we can meet some inadequate response to antigen. This means that some breaks in these immune synapse, in these steps of specific immune response. And here you see the various hypersensitivity stages were originally that classified into types 1, type 4 by Gel in Coops, and this classification remains broadly useful nowadays. Here you see type 1 hypersensitivity, it's also called anaphylaxis and allergy. Type 2 hypersensitivity, another term antibody-dependent cellular cytotoxicity. Immune complex diseases causes by type 3 hypersensitivity, and we have type 4 hypersensitivity, and also the type called like delayed type hypersensitivity. So hypersensitivity type 1, also called lead to allergy and anaphylaxis reaction. This is immediate hypersensitivity type, like immediate reaction to the introduction of antigen, which is accompanied by specific symptoms like swelling, convulsion, narrowing of blood vessels, nausea, heavy breathing, and loss of consciousness. And like you see, the first aid is normally knowing for all of us. List of allergens is given in this slide. This classification based on portal of entry and includes inhalants, antigens, ingestants like by food, injectants by different drugs, vaccines, serum, and contacts by cosmetics, some detergents. The good example by ingestants, allergens by food, milk, shellfish, nuts, the most common foods that cause allergies. Symptoms, like we said, include these breathing problems, swelling around the mouth, and the symptoms can be severe, and the symptoms are very fast, leading to activation. Avoid, we need to avoid these fatty foods altogether in case if we allergic to this one, or but it if exposed, this can be life-threatening, and we have first aid. This is administration of adrenaline to reveal smooth muscle spasm that we can breathe correctly. So the hallmark of type 1 hypersensitivity reaction is production of immunoglobulin E by synthesized B cells following and contact with allergen with induced mast cell degranulation. Like we said before, that in case with immunoglobulin E, it can be non-specific mechanism of activation. The pharmacologically active mediators released from these granules of mast cell degranulation can be histamine, prostaglandin, and leukotrienes, and these granules cause vasodilation, vascular and smooth muscle contraction, and increased vascular permeability. These changes ultimately lead to localized response. In case with localized, this called atopy, or in case with systematic response, this is called anaphylaxis shock. Type 1 hypersensitivity reaction occurs through two phases: the sensitization and effector phases. Both phases occurs in interval of 2 weeks. In this slide, you see the mechanism of type 1 hypersensitivity reaction. This is a sensitization phase. Firstly, the same in specific immune response. Firstly, other antigen-presenting cells meet with naive T cells and activate by MHC complex. In case here in MHC2 complex, activate T helper cells. In case with this one, this is T helper cells number 2. T helper cells release some cytokines, and these cytokines can activate B cells. B cells after proliferate and differentiate into plasma cells that synthesize a specific type of immunoglobulin E. Immunoglobulin E can bind to mast cells by Fc region, and these mast cells start to be sensitizing mast cells. And also these mast cells, this activation of mast cells lead to their degranulation, like we talked before, that this degranulation release inflammatory molecules that can result in allergy symptoms, anaphylactic symptoms. In case with effector phase, when the same allergen antigen is induced, this is in case with shocking dose, this allergen is introduced subsequently to our body. It directly encounters with the FAP regions of immunoglobulin E coated on mast cells, and this is rapid methods, and because of this is anaphylactic shock. Not first exposure, because you need time for specific immunity. In case with this effector phase, this immediately lead to release of these inflammatory molecules. So go to the next type. This antibody mediated type 2 of hypersensitivity. In this case, immunoglobulin M we see, and immunoglobulin G antibody specific for cell surface or extracellular matrix antigen can cause tissue injury by activating the complement system and recruiting by this activation inflammatory cells and by interfering with normal cellular function. This means this is like normal function of these antibodies. They activate phagocytosis line of our innate defense, but in this case, this is component of this autoimmune disease and this antibody-dependent cellular cytotoxicity. They invade these combined activities, a lot of activities of complement system and antibodies, and this hyper activity leads to these disorders. So, in case with classical case, antibody mediated antibody-dependent cellular cytotoxicity, this involves some NK cell, natural killer cells, and these natural killer cells also have specific Fc in this case here. Fc fragments, Fc receptors that can be activated by immunoglobulins. This is called CD16 receptors. And these CD16 receptors, Fc receptors, recognized by Fc fragments of an antibody, which can bound to pathogen infected target cells. Once these Fc receptors bind to Fc region of immunoglobulin G, natural killer cells start to be activated, and because of this, in case with type 1 hypersensitivity, these activated NK cells need produce some cytokines, and these natural killer cells release cytokines such as inflammatory cytokines like interferon gamma, and cytotoxic granules contains perforin and granzymes that enter the target cells and promote cell lysis by apoptosis. This is similar by cytotoxic C cells, like we called about NK cells in non-specific immune response. But this is inadequate reaction of NK cells in inappropriate way, okay? And also we have another type of this cytotoxicity. This is called antibody-mediated cellular cytotoxicity mediated by phagocytes. Here, this is classical activation of antibody-mediated cellular cytotoxicity in case with NK cells. This is spontaneously activated on NK cells. In this case, this is more complex mechanism, because also the Fc region of antibody, of immunoglobulin M antibody, immunoglobulin G antibodies, can activate complement system, activate the classical pathway that we mentioned before, that Fc fragments of immunoglobulin can bind with C1q components of classical pathway. And this activation of classical pathway by Fc fragment of immunoglobulin and C1q components lead to host cell injury, which is mediated by following mechanism. This is not the classical mechanism. This can be complement dependent cytolysis, like the same with complement system activation. Information of membrane attack complex formed by activation of classical pathway can produce spores in cells that lead to lysis of target cells. But in this case, this is not microbe cells, this is our cells. And also another mechanism here can be by opsonization. Byproducts of complement pathway, if you remember, we have C3b and C4b complements, and these complements act as opsonins, and they deposit on the target cells. And these phagocytosis, our specific phagocytes such as macrophage and neutrophils can engulf these cells with opsonins, with C3b and C4 coated target cells via complement reaction, and this also can lead to phagocytosis. This antibody-dependent cellular cytotoxicity and antibody-dependent complement-mediated type 2 hypersensitivity are observed, like we said before, in different combination of many autoimmune disease. And here you see various clinical conditions on this slide, and you see that clinical manifestation also, which diseases we have in this case. But I want to share with you some example. One example about autoimmune hemolytic anemia, or in case with clinical, we call this one thrombocytopenia. All these result due to the production of autoantibodies, self-antibodies, to the individuals of membrane antigen of erythrocytes, And for example, antibodies specific for the nicotinic acetylcholine receptors cause functional abnormalities that lead to myasthenia also gravis disease. In this case, you see antibody down modulate receptors, and because of this we have these specific disorders that can lead to paralysis and muscle weaknesses. So our third type of hypersensitivity this is called immune complex hypersensitivity. This is a result of excess formation of immune complexes. I need to remind you that immune complexes, this is complex of antigen and antibody. These immune complexes can initiate inflammatory response through activation of complement system, leading to tissue injury. The same, inadequate reaction, inadequate hyperactivation of this complement system, hyperactivation of all this can cause some systematic or localized reaction. The localized and systematic reaction, the major mechanism of tissue injury in immune complex diseases is inflammation within the walls of blood vessel, resulting from complement activation and binding to leukocytes Fc receptors to the antibodies in the deposited complexes. And if you remember from previous slide, we have the same activation, and also in this case we have lead roles of anaphylaxis toxin, these components of complement system C3A, C4A, C5A components production, mast cell degranulation, macrophage activation, platelet aggregation, and also influx of neutrophils all make their contribution. This means that inflammation in blood vessels, inflammation within blood vessels, and all cells, all inflammatory releases cytokines can go through to this local system. In good example, or it can be systematic reaction when we don't have specific localized. For example, with some localized reaction, this is glomerulonephritis, and you see different types of glomerulonephritis. First one, this is A, glomerulonephritis induced by antibody against the glomerular basement membrane. Guts-Pasteur syndrome is called. And this light micrograph shows glomerular inflammation and severe damage. And immunofluorescence shows linear depositive of antibodies along the basement membrane. In case with immune complex mediated glomerulonephritis, this induces by immune complex deposition, systematic lupus erythematosus, and as you see, the light microscope shows neutrophilic information because, like we talked before, a neutrophil, anaphylotoxin, mast cell degranulation, macrophages activation make all contribution in this localized tissue. And here electron micrograph shows granular, not linear, but granular deposits of antigen antibody complexes along the basement membrane. This is differentiation, and based on this you can go to the symptoms. And the last one, and most interesting maybe, dilated type for hypersensitivity. This hypersensitivity differ from other types in various ways. First one, this is delayed types. This means that these types occurs after 48-72 hours of antigen exposure. Also, this is cell mediated by specific cells, mediated like T helper cells. Also, tissue injury occurs predominantly due to activated macrophages. And this kind of hypersensitivity is encountered in many allergic reactions to infection agents from the contact dermatitis to transplant rejections. And this hypersensitivity, because this is delight types, these types occurs in two phases. First phase is this is sensitization phase, and second one effector phase. During the sensitization phase, the antigen presenting cells like macrophages, dendritic cells process and present the antigenic peptides along with MHC2 complex to helper T cells, to T0 cells, and T0 cells activate T helper cells. Most T helper cells in this case are Th1 cells. In case with effector phase, these T helper cells, Th1 cells, they have subsequent contact with the antigen and secrete a variety cytokines, and these cytokines can attract and recruit inflammatory cells, again such as macrophages, at the site of reaction. I need to mention that interferon gamma is the key cytokine of type 4 reaction. It activates by resting macrophages into activated macrophages, and these activated macrophages start producing a lot of cytokines. Perhaps the most known example of dilated type hypersensitivity, this is MAN2 reaction obtained by injection of tuberculin. I need to remember you that tuberculin, this is extract of Mycobacterium tuberculosis into the skin of individual from a previous infection with Mycobacterium had induced a state of cell-mediated immunity. The reaction is manifest by induration with redness and swelling at the site of challenge after 48 and peaks in 2 days. And here you can compare first day and second days. So, immunological tolerance is a responsiveness to antigen induced by exposure of specific lymphocytes to that antigen. Tolerance to self-antigens is fundamental property of the normal immune system, and the failure of this self-tolerance leads to autoimmune disease. Also, disorders caused by abnormal immune responses are called hypersensitivity diseases, and pathological immune responses may be autoimmune response direct against self-antigen or uncontrolled and excessive response to foreign antigen. Hypersensitivity diseases may result from antibodies that bind to cells and tissues, type 2 hypersensitivity, circulating immune complex that are deposited in tissues, type 3 hypersensitivity, or T lymphocytes reactivate with antigens in tissues, type 4 hypersensitivity, or immediate hypersensitivity, type 1 reaction as the cause of allergic disease. This is our all information about different disorders that can occur in our specific immune system. In case with autoimmune disease, the same. This is breaks of tolerance. In case with hypersensitivity, this is inadequate immune response. So, next our lecture part is getting to know therapeutic approaches for immunological disease. First of all this started in 1890. Baron Kitazato discovered the therapeutic effect of antitoxic diphtheria and tetanus serums obtained by immunizing animals with living microbes, and later with the exotoxin. Also, Baron received Nobel Prize for this discovery, bearing joint forces with industry to produce large doses of antisera for human use, thus embodying the translation immunologists with great interest in medical application. Serum therapy was more than just a curative method. It also provided supportive evidence for the idea that the cause of infections disease is highly specific and that this specificity is linked to toxin produced by etiological pathogen, specific pathogen. As corollary, the cure of specific disease was accompanied by specific poison averting agent. In case this one, we call this one antitoxic, which circulates in blood and can offer specific protection against the toxin in other individuals. Nowadays, we call this phenomena is passive immunity. Artificially acquired passive immunity is a short-term immunization achieved by the transfer of antibodies, which can be administered in several forms, as human or animal blood plasma, serum, pooled human immunoglobulin for intravenous or intramuscular use, and as monoclonal antibodies. This is also a potential risk for hypersensitivity reactions and serum sickness, especially from gamma globulin or non-human origin. Passive immunity provides immediate protection, but the body does not develop memory. Therefore, this passive immunity, this immediate protection, can lead that patient now at the risk of being infected by same pathogen later unless they acquire active immunity by the vaccination. The following immunoglobulins are the immunoglobulins currently approved for use for infectious disease prophylaxis and immunotherapy in the United States by FDA. Passive transfer, it can be used to prevent diseases or used prophylactically in case of immunodeficiency, some stages such as gamma-globulinemia. It is also can be used in the treatment of several types of acute infections, like this one, and to treat poisoning. For example, treatment of wound or foodborne form of botulism, infant botulism is treated with human botulism immunoglobulin, and commercial name, this is baby big globulin, baby deep medicine. Let's describe one unique form of treatments, immunoglobulin, a monoclonal antibody. When an antigen having multiple epitopes enters the body, each epitope may stimulate one clone of B cells, producing one type of antibody. Hence, the result of antibody mixture present in serum, it can be said that these antibodies to be polyclonal, which means contains mixture of antibodies derived from different clones of B cells. However, when only one clone of B cell is stimulated by single epitope of antigen, and then only this B cell, one clone of B cell, is allowed to proliferate and produce antibodies, such antibodies are referred to as monoclonal antibodies. Monoclonal antibodies are produced by hybridoma technique developed by Coyle and Milstein in 1975, for which they were awarded Nobel Prize in 1984. Monoclonal antibodies have become a viable tool for both diagnosis and therapy of several diseases. However, monoclonal antibodies are more expensive to manufacture than small molecules due to the complex process involved in the general size of molecules, which increase production costs. Several clinically important monoclonal antibodies are used in the treatment. Most therapeutical applied treatment you can see use in the treatment in cancer and inflammatory diseases. This is the more antibodies, monoclonal antibodies that we can provide for this one. For example, trastuzumab, which is sold under the brand name Herceptin, is monoclonal antibody used to treat breast cancer. While this therapy really has shown great efficacy, it comes at a higher price, at a higher cost. Approximately one course can be almost $70,000 for full treatment course. And this is difficult hybridomas techniques, as the methods relies of using B cells from immunized animals. Typically it can be mouse, with immortal myeloma cells, mutant myeloma cells, and growing in cells under conditions in which the infused normal and tumor cells cannot survive. For this purpose, we can use specific media, this media and this method called hot selection. The selective culture media is called hot medium because it contains hypoxanthine, aminopterin, and thymidine, EHAT. This medium is selective for fused only hybridoma cells, immortal myeloma cells, and B cells from mouse spleen. In case with infused myeloma cells, they cannot grow of this media because they lack hypoxanthine guanine phosphorybolase transferase, and those cannot replicate their DNA. Only fused hybrid cells, referred to as the hybridoma cells, are able to grow in this medium because the spleen cell partner supplies this hypoxanthine guanine phosphorybasyl transferase, and this myeloma partner has traits that make it immortal like cancer cells. Each hybridoma in this case produce only one type of immunoglobulins derived from one B cells from spleen of these mice. In this case we have polyclonal B cells and polyclonal antibodies. The antibodies later created by many hybridoma clones are screened for binding to antigen of interest, and the clone with desired specificity is selected and expanded. The products of these individual clones are monoclonal antibodies. Okay, we mentioned artificially also acquired immunity, and this is two types: passive artificial acquired immunity and active. In case with active immunity, this is vaccination, immunization, or process by which individual's immune system becomes ready against an infection agent called immunogen. Immunization happens in various ways, both in the wild and as done by human effort in healthcare. Natural immunity is gained by those organisms whose immune system succeed in fighting off with previous pathogen. The first vaccine created and used, the first vaccine used was by Edward Jenner, a vaccine against smallpox, a particularly dangerous fatal disease that spread epidemically and destroyed entire population. And in this year, 1789, Edward Jenner used a vaccine against smallpox. He often called the father of immunology. We all know maybe this phenomena. Experiment on May 14, Jenner tested his hypothesis by inoculation James Phipps, 8-year-old boy who was the son of Jenner's gardener. He scraped pus from cowpox blisters on the hands of Sarah Helmes, a milkmaid who had caught cowpox from a cow called Blossom, whose hide now hangs on the wall of George's Medical School library. Phipps was the 17th case described in Jenner's first paper on vaccination. Jenner inoculated Phipps in both arms that day, subsequently producing in Phipps a fever and some anæmias, but no full-blown infection. Later, he injected Phipps with various material, the routine method of immunization at the time. No disease followed. The boy was later challenged with various material and again showed no sign of infection. No unexpected side effects occurs, and neither Phipps nor any other recipient underwent any future breakthrough case. In the second half of 19th century, the scientific principles of obtaining vaccine were developed by Louis Pasteur. He received vaccines against anthrax, rabies, cholera, and suggested that the terms vaccination be named after Edward Jenner. Application of Louis Pasteur methodology made it possible to create vaccines against many viral and bacterial diseases that we use also nowadays. So, the vaccine is biological preparation, biological medicine that provides active acquired immunity to a particular infectious or malignant disease. And ultimate goal by WHO, this is eradicate of this specific disease by vaccination. There are several types of vaccine, including live attenuated, inactivated vaccine, whole cell inactivated, subunit, toxoid, recombinant vaccines, viral vector vaccine, and modern messenger RNA vaccine. Traditional vaccines consist of entire pathogens that have been killed or weakened so that they cannot lead and cannot cause disease. They may contain live viruses, live bacteria that have been attenuated, in case this term means you weakened or altered to not cause illness. Also, they may contain inactivated or killed organism, microorganisms or viruses. They can contain inactivated toxins for bacterial diseases where toxins generated by the bacteria and not the bacteria themselves cause illness. Exotoxins, in case with diphtheria, for example. In case with tetanus, for example, or merely segments of the pathogen. This includes subunits vaccine, conjugated vaccine, some unique units of these bacteria that can lead to immunogenicity and antigenicity. So, whole pathogen vaccine can elicit strong protective immune responses. Many of the vaccines in clinical use today fall into this category. Scientists first described the ability of inactivated or killed microbes to induce immunity in the 19th century by Louis Pasteur. This led to the development of inactivated vaccines, which are produced by killing the pathogen with chemicals, heat, or radiation. One contemporary example such a vaccine, this is Havrix, inactivated vaccine against hepatitis A viruses that was developed in 1985. Advances in tissue culture techniques in the 15 years of 20th century enabled development of live attenuated vaccines, which contain a version of the living microbe that has been weakened in the laboratory. Good example, MMR vaccines, measles, mumps, and rubella vaccines is good example of live attenuated vaccines. These vaccines elicit strong immune responses that can confer lifelong immunity after only one dose. Also, in the 1970s, advances in laboratory techniques ushers to us era of genetic engineering. A decade later, recombinant DNA technology, which enables DNA from two or more sources to be combined, was harnessed to develop the first recombinant protein vaccines. And used nowadays, these vaccines, there can be hepatitis B vaccine. The vaccine antigen is hepatitis B viruses protein produced by E cells, which is a genetic code for viral protein has been inserted. Vaccines also based on messenger RNA, intermediate between DNA and protein, also were developed. And great example can be vaccines against coronavirus that we use in this century. Rather than delivering DNA or mRNA directly to cells, some vaccines use harmless viruses or bacterium as a vector or carrier to introduce genetic material into our cells. And in case this viral vector vaccine, also some example of viral vector vaccines can be vaccines against coronavirus. So there's several types of vaccines, and vaccines need to be safe and effective. Certain ingredients help keep vaccines safe from contamination and toxins. Other, like stabilizers, help vaccines stay effective for a long time, and creating safety in vaccine production and usage are reference to some domains. In this case, you see this on the slide. This vaccine need to be standardized, and if you not standardize this vaccine, you can't use this in commercial usage. So some examples, some specific example of classical vaccine type. This is live vaccine, live attenuated vaccines. Like we mentioned before, live attenuated vaccine contain live pathogen from either bacteria or viruses that have been attenuated or weakened by heat, radiation, or chemicals reagents. In the lab, generally the virus is passed through many generations of cells to pick up genetic mutation which weaken it. Such vaccine is strong enough to trigger immune response, but too weak to cause the disease. Very great benefits from this vaccine because they contain this live pathogen. The immune system react really very well and to them, and they typically remember this pathogen for a very long time. We create long life immunity. Examples, like we said, MRR vaccine, measles, mumps, and rubella vaccine. Another classical vaccine type, this is inactivated vaccine. Inactivated vaccine take a live pathogen and inactivate it, like kill it by heat or chemicals, also components. When the vaccine is then introduced to a human through a shot, the inactivated pathogen another great example of classical vaccine type, this is inactivated vaccine. You take a live pathogen and inactivate, like kill this pathogen by heat or chemicals, by chemicals reagents. When the vaccine is then introduced to humans through a shot, the inactivated pathogen is strong enough to create immune response, however, also incapable of causing some disease. But in case with this one, you require for multiple doses to order to build up immunity and offer full protection. But also, compared to live attenuated vaccine, inactivated vaccine can be mass produced and are relatively inexpensive to make. And some great example of inactivated vaccine can be polio vaccine. Also, I want to add some information, and by the way, despite of this bearing honors that he received, he was not fully satisfied with passive vaccination that he created. In order to neutralize the diphtheria toxin, Beren generate also antigen antibody complexes, which stimulated production of toxin-specific antibodies in the immunized host. This was an important but still suboptimal start toward active vaccination against bacterial toxin. For this one, it was the French researcher Gaston Ramon who ultimately introduced detoxification by formaldehyde for low-cost production of safe vaccine against diphtheria and tetanus. And also he adds some aluminum hydroxide as adjuvant for potent immunization, for big immune answers to this vaccine. Nowadays, thanks to this tool, we have the type of vaccines that called toxoid vaccines. You see the mechanism how you can create these toxoid vaccines. You use inactivated toxin to target the toxic activity created by the bacteria rather than targeting the bacteria itself. The goal of toxoid vaccines is to give people a way to neutralize those toxins with antibodies through vaccination. You create antibodies against this toxin before these bacteria start producing this toxin. Toxoid vaccines especially good preventing certain toxin-mediated diseases such as tetanus, diphtheria, and pertussis. But we have some minus because you create antibody against toxins, and booster shots are typically recommended every 10 years or so. And this example, diphtheria and tetanus toxoids vaccines. And you need to remember that each country has recommended immunization schedule, and you also can maybe look through at immunization schedule in your country. Most parents vaccinate their children according to CDC recommended immunization schedule, protecting them from 14 potentially serious disease before their second birthday. Vaccination to vaccinate children on time protects them and anyone around them with a weakened immune system. It's safe because hundreds of the country's top doctors, public health professionals, and scientists designed the schedule to ensure it is safe and effective. And in this recommended, you see that this recommended immunization schedule includes a range of recommended ages for all children, a range of recommended age for catch-up vaccination in case if you miss some vaccination, a range of recommended age for certain high-risk groups in cases HIV positive people or in case with weakened immunity, soft autoimmune disease, and so on. So, vaccine is biological preparation that improves immunity to a particular disease, and you see that thanks to these vaccines we almost forgot about these 14 diseases. And I need maybe to mention that for example measles and polio, this is very dangerous diseases, and nowadays almost we never mentioned if in clinical cases. So be safe through the vaccination. This is good. Thank you guys for your attention, and this is some resume for our lecture.